In this video, we will discuss about effect of T cell differentiation on FAS-DNR modified T cells efficacy. So, for those who are not aware about DNR or who has not gone through of who has not gone through the previous videos, this DNR is dominant negative receptors. So, the approach this paper uses is they overexpress this mutant FAS on the T cells so that it can uh, reduce the effect of the FAS ligand interact with the wild type FAS that will trigger apoptosis. So this the result of this approach has been discussed in our previous videos. You can go through those videos to check out how those approaches effective. So here what the researchers doing is they wanted to check whether this T cells differentiation has an effect on the FAS-DNR modified T cells efficacy on the tumors or not. So in order to nullify the effect of these T cells differentiations, so how the researchers do this studies is that they nullify the T cell differentiations and then check the effect of the wild type FAS, the T cells with the without any construct, and T cells with this mutant FAS construct. So how they nullify the T cells differentiation effect is that just before they transduce the T cells into the mice, they sorted this T cells populations that are CD62L positive, Thi 1.1 positive, and CD44 positives. And after injections or before they transduce these T cells into the mice is already been transduced with the virus tumors, which is already been transduced with the tumors. And after they inject this sorted T cells populations, they also inject the IL-2 for 3 days so that T cells remains active, and thereafter they check the tumor areas at different time intervals at different days, and what they are able to see is that where there is no T cells, the tumor area increases uh, in a short duration of time, whereas where there is empty construct is transduced into the T cells, they can see a increase in the tumor area takes some more time compared to the conditions where no T cells are transduced and no T cells are injected into the mice. And in the case where they inject the modified T cells that has the FAS-DNR or the intercellular domains of the FAS signalings has been truncated, they are able to see the tumor area increases, takes a longer time. That is, even after 40 days, they see a tumor area increase at a very small rate. And when they check the survival rate of these conditions, they are able to see that when there is no T cells are injected into the mice dies in a short duration of time, whereas where they introduce the uh, T cells with the empty construct, they are able to see increase in the survival rate. But when they check the survival rate of the mice that are injected with the modified T cells, that is the intercellular part of the FAS signaling has been truncated, they are able to see a maximum survival rate. So these results are also in line with the results that we have seen in the previous videos, that when the T cells are transduced with this modified FAS DNR construct, or in shorter term we can say that when the intercellular FAS signalings um, has been truncated, we are able to see similar results, that is increase in the survival rate. And here where when we take the same populations of these T cells and but use the different CAR construct, we see the same effect in terms of the survival rate. So this indicates that the T cells differentiation has very minimum effect or almost negligible effect on the T cell modified the FAS DNR modified T cells efficacy. I hope you understood the results that are presented here. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.